Hi, my name is Simon Larsen, and this is a short video tutorial on how to use CoNVaQ. CoNVaQ is a web tool for performing copy number variation-based association studies. On my screen here, you can see the CoNVaQ front page. You can find the website at convaq.compile.edu.dk. On the top of the page here, we see the main menu. Under Guide, you'll find a written tutorial covering the same steps as I'll cover in this video. If we go down to File Format Specification, we can also find a description on how to structure our data in order for it to work with CoNVaQ. Let's scroll back up to the top of the page. Select Get Started from the main menu in order to start your analysis. The first thing we need to do is to select which species and reference genome we are working with. I'm going to select Homo sapiens with the HC18 assembly. Below, we see two panels named Group 1 and Group 2. These are the two groups we want to compare in our analysis. For this example, we're going to compare two groups of CNV calls from HPV positive and HPV negative samples. Under Name, we can name the first group HPV positive. Then click Browse and select the file containing the CNV calls. Similarly, we name the second group HPV negative and select the corresponding file. Once everything is ready, we can click Upload Data. When the upload is complete, we are brought to the analysis page. Under Summary, we see some basic statistics for the dataset. We recommend you double check that these numbers all look correct in order to verify that your data was formatted correctly. Under Model, you can choose between two models for significance testing: the statistical model and the query-based model. We're going to start with the statistical model. In the Options panel, we can set the parameters for the model. Under p-values, you can choose between using a one-tailed or a two-tailed test when computing the p-values, depending on the hypothesis you're testing. For this analysis, I'll choose a one-tailed test. You can also set a p-value cutoff. All CNV regions with a p-value greater than this threshold will be disregarded. I will keep this value at 0.05. Under Merging Regions, we can choose whether to merge significant regions into a single region if they are adjacent within some number of base pairs. I will keep it at the default of merging regions only if they are strictly adjacent. Once everything is done, we click Submit in order to submit the query. Depending on the size of your dataset, this might take a few minutes. Once the computation is done, the results will show up in the table under Results. Each row in the table represents a CNV region with significant p-value. The first few columns here tell us the position and length of the region. The event column tells us what type of event we found. For instance, in the first region, we detected a loss event. Here we have the p-value and q-value. The remaining columns tell us the frequency of each CNV type for the two groups. In the first row here, we see that 21% of the samples in the HPV positive group have a loss in copy numbers in this region. For the HPV negative group, this number is 0%. For some regions, the frequency is represented as a range. This happens when two or more regions with different frequencies have been merged. For instance, in the second region here, we see that the frequency of loss events in the HPV positive group ranges from 29 to 36%. If we click the magnifying glass in the leftmost column, we can inspect the individual regions. Clicking this button brings up a new window. Under Summary, we have the same information as in the results table, shown in a more readable way. Under Sample States, we can inspect the individual state of each sample. Click the name of a group to expand the panel, showing the state of each sample in this group. Under Genome Browser, if we click Show, we can also open this region in the UCSC Genome Browser. Click Close to close this window and return to the page. If you want to save your results, you can use the three buttons located above the results table. The Copy button will copy the contents of the table to your clipboard. The other two buttons will allow you to save the table either as a CSV file or an Excel table. Let's try the query-based model instead. First, select the query model. Say we want to search for loss events in the HPV positive group. In the HPV positive group, I select at least 20%, equal to, and loss. And in the HPV negative group, I select at most 5% equal to and loss. This query specifies that we are looking for regions where at least 20% of the HPV positive samples and at most 5% of the HPV negative samples have a loss in copy number. The other options below here are the same as for the statistical model. I will leave these as default. When we click Submit, we obtain the results in the table below. This table is similar to the one we get with the statistical model, except no p-values are computed. The query-based model allows for a lot of flexibility. For instance, we could change the query for the HPV negative group to also disallow other types of variations as well, by sending it to at most 5%, not equal to, and normal instead. This means we now only allow regions where at most 5% of the samples in the HPV negative group have any kind of variation. We can also restrict this even further by setting the frequency to 0%. For large data sets, the Q-value computation may take a long time. When this is the case, we recommend you switch off the Q-value computation while playing around with the queries. Then, when you have found a set of interesting regions, you can switch it on again and repeat the query in order to obtain the Q-values. Once we have obtained a set of CNV regions, we can perform a basic enrichment analysis. In the results table, you can select one or more regions by holding down the Control key and left-clicking on the rows. Selected regions will be highlighted with blue. Once you have selected a set of regions you want to analyze, click the Analyze Selected Regions button below the table. This will bring up a new window. After a moment, a table will show up containing all the known genes that are overlapping with one of the previously selected regions. Here we get the name of each gene, the biotype, and a short description. You can also click the Ensemble Gene ID to look up the gene on the Ensemble website. In order to do gene set enrichment analysis, select the Enrichment tab from the top. In this drop-down menu, we can select what type of terms or classes to look for. We can choose between Go terms for function, process, and cellular location, as well as Panther protein classes and pathways. For this example, I will select Protein class. Click the Search button in order to start the analysis. After a moment, a table will appear below. In this table, we have the ID and name of each class, as well as a p-value and a list of genes matching this protein class. That concludes the tutorial. We hope you'll find CoNVaQ useful, and thank you for your attention.